All right, listen up.
We are rolling.
My turn. Stop talking. Start listening. Huh? You like that?
I'm so scared I'm gonna kill this.
We learned about glycolysis last time, so we're basically on page 140, section 8.3. We have 2 days to figure out what happened to the glucose. The test is about 2 weeks. 2 weeks? What movie? 2 weeks. The Money Pit.
I love that movie.
Great movie. Okay, so let's kind of this is kind of review what happens in glycolysis. Guess what this hexagonal shaped molecule is?
Glucose.
That's glucose. Let's watch what happens to it.
Close, yeah.
Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins.
That was a good overview of what happened in glycolysis. Basically, it's taking glucose, this hexagonal molecule, which I'll represent now as this, and breaking it apart.
Why does that happen?
Well, it happens because the whole point of respiration is to tear glucose apart and get energy out of it. And the energy is used to do what?
Make ATP.
Make ATP. To take ADP and P and put it together to make ATP. So we got to tear up glucose. That's the whole point here, to get the energy out of the bonds in this glucose molecule to make ATP. And so the first step in that, do y'all remember the 4 steps? Good people can achieve. Good people can achieve. That doesn't make sense. Good people can excel. What do those stand for?
Glycolysis, preparatory reactions.
Citric cycle, electron transport chain.
Glycolysis, prep reaction, citric acid cycle, electron transport chain. And G, glycolysis, that happens outside the mitochondrion. That happens first. As soon as the glucose enters the cell has enzymes that can tear it apart. And some of the energy, whenever you take a big molecule and you tear it apart, some energy comes out. Some electrons come out. Some protons come out. And those electrons and protons are picked up by electron carriers like NAD, will pick up hydrogen and pick up some electrons and shuttle them off into the mitochondria. I like to think about, have you ever watched a movie with, what's the big monster's name from Japan? Godzilla movie. Have y'all ever seen that? And Godzilla will pick up a car and just rip it apart. And whenever you take something large and rip it apart, stuff comes flying off of it, like electrons and protons. And those got to be picked up and taken somewhere. This area of the class doesn't seem to be following me at all. It's kind of like, just, if you could just sort of. The rest of y'all doing pretty good, but just kind of, I don't know, what's
That's exactly what I was going to say.
what is the deal? So, glucose gets torn in half, and what are we left with? What are these two new molecules called at the end of?
Pyruvate?
Pyruvate.
Pyruvate. They're called pyruvate. Pyruvate. Also known as pyruvic acid. Y'all, if you ever see 8 on the end of a word, you can substitute ic acid for 8. So pyruvate can also be called?
Pyruvic acid.
Citrate can also be called?
Citric acid.
Y'all are good?
What does pyruvate do?
Pyruvate then will enter the mitochondrion. Along with the NADHs that were formed, they'll enter the mitochondria.
What's G3P?
And then what?
G3P is a molecule of glyceraldehyde 3-phosphate.
That's in the Calvin cycle.
Yeah, that was back here. That's what we formed at the end in the Calvin cycle in photosynthesis.
Okay, so the glucose gets ripped apart by what?
By special enzymes that are just floating around.
Okay, and then it goes into the...
Yeah, after we rip apart the glucose, we no longer have glucose. What do we have?
Pyruvate.
And that enters the...
Mitochondria.
And now we go into P, C, and E.
That's all you need to know.
Glycolysis is outside of the mitochondria.
Glycolysis is outside of the mitochondrion. Very good lead. Very good lead. Did you get that, Emily Kay? It was a great joke.
Hilarious.
You didn't even react.
I know, I was focused.
Okay. No, that's not all you have to know, because whenever we rip apart something, stuff flies off, like protons and electrons, and those will get picked up. They don't just float around. They get picked up by electron carriers, like NAD picks up the electrons and protons, and then takes them into the mitochondria.
Okay, so...
Doesn't FAD do that too? But...
FAD will do that later. We'll see FAD a little bit later.
So it comes pyruvate, and the pyruvate goes into the mitochondria.
There you go.
That's all glycolysis.
Well, it actually occurs in 10 different steps. You want to see the 10 steps?
Oh, good.
There it is. That's all glycolysis is.
Do we need to know, we just need to know that it creates 4.
It makes 2 ATP net.
Yeah.
It creates 4, but it uses 2 at the beginning, so that you got a net gain of 2 ATPs, and you gain 2 NADHs come out of it, and pick up protons and electrons and enter the mitochondrion, and you get the 2 pyruvates.
Whenever the screen's on this thing, it makes a rainbow.
Yeah, it does. It's because the frames per second on that are different than the frames per second on the projector.
It's pretty.
It's a technology.
You can adjust that, can't you? If you go on settings on your computer, you can change your frame rate, can't you?
What is that?
Isaac, that is so awesome. We need to figure that out, you and I. Let's do that. I'll give you some extra credit if you can help me.
Oh, do it.
All my friends ready.
Isaac beat you to it. Sorry.
I can't.
Yeah, what was your question?
So 2 NADHs are made.
Yes. 2 NADHs are right. Other questions?
So 2 ATP net gain, 2 NADHs, and
2 ATP, 2 NADHs, and 2 pyruvates. And and all that came from what?
Glucose.
A glucose.
It's really cool.
Okay?
Is that picture in our book?
Absolutely.
139.
139.
So it gets 2 NADH, 2 pyruvate, and 2 ATP.
There you go. Do it, Emory.
Got it.
Are you going to go to Emory?
So way.
What's that?
Why? You have a bunch of
Emory University.
name on it.
Are you going to go to the college, Emory?
No way.
My parents went to Emory.
No.
So did my grandparents.
Nice. Is that why you were named Emory?
No, it's named after my great grandfather.
His name was Emory?
Yes, sir.
And his kids went to Emory?
Yeah, different.
What are the odds of that? Okay, sorry, I'm getting sidetracked. All right. So, let's talk about this process called fermentation. Have you ever heard of that?
Yeah, like alcohol, right?
Yes, it's how alcohol is made. And that's an important concept to be discussing after this last weekend.
Right.
Now,
Right.
it was homecoming dance for the viewers at home. The pyruvates will only go into the mitochondrion if oxygen is present. Oxygen is very important for the further breakdown of these pyruvates. These pyruvates will go into the mitochondrion, and oxygen that you breathe, O2, remember O2, will also go into the mitochondrion. But if oxygen is not present, the pyruvates cannot be broken down. And the pyruvates will build up inside the cell. Now, the pyruvates are very toxic and can kill a cell.
So why is it going into the cell?
Why do they want them then?
Well, because it's high energy, and if it goes into the mitochondrion can break it down further and make a bunch of ATP.
How does it kill the cell?
Toxic?
It's not toxic if it goes into the mitochondrion and is broken down further.
Why does it need to be toxic? Like, why do
It just
they?
is.
Britney Spears.
How does it kill the cell?
Why would?
It's like a poison to the cell. I think it interferes with enzymes that are in the cell, but
So it's an
I'm not
enzyme?
exactly sure. I learned that something like
Like an
25
enzyme
years ago.
inhibitor?
What's that?
Enzyme inhibitor?
Yeah, probably something like that.
Okay.
Why wouldn't oxygen be in the mitochondria?
Ah!
Because it's aerobic.
That's what I was going to get to. Sometimes a cell can be left without enough oxygen. For instance, after you run a sprint, you're sprinting so fast, your cells are working so hard that you can't get enough oxygen into your blood. For the first 20 seconds of the sprint, you got oxygen, but you just can't breathe fast enough to supply those cells with enough oxygen. And you start to become, the cell starts to not get enough oxygen. And so you start to build up this pyruvate. And the cell does something quickly. It goes, hey, this pyruvate is toxic. I am going to make it less toxic. And the cells in your body will change the pyruvate to a different chemical called lactate.
Oh, so
And the way it does that is the NADHs that were produced earlier come back and change it a bit. So they give back their hydrogens and their proton and their electrons. They give the protons and electrons back, and it changes it a little bit from pyruvate to lactate. And lactate is also called
Lactic
acid.
acid.
lactic acid. And lactic acid is what you feel burning your muscles when you're running a sprint or lifting weights real fast. Your muscles start burning. That's lactic acid. When you finally get enough oxygen, when you're breathing, and after the sprint you stop and you're going, and you're pulling in as much oxygen as you can, and that oxygen is going into your mitochondrion, and then finally, after you breathe hard, there's enough oxygen there, then the lactic acid can be converted back to pyruvate by the NADH picking up the hydrogens and the electrons, and now we got pyruvate again, and then the pyruvate will go into the mitochondrions now that there's enough oxygen present and be broken down.
So
That
one,
is fermentation.
Fermentation is the process of producing the lactic acid.
I don't understand how
So if
lactic
the-
acid was produced.
so if you're not getting enough oxygen, the pyruvate will just sit there.
Yeah, there's no oxygen inside the mitochondria. The pyruvate
If there's
will
no
sit
oxygen, the pyruvate's sitting there, and the NADHs that were produced have to come back and give back protons and electrons and change the pyruvate to lactate.
So, like, if you're just breathing normally, then it's just going to be sitting there?
Now, if you're breathing normally, just in normal circumstances, there's enough oxygen.
Okay, so if you're not breathing.
If you stop breathing,
If you're underwater.
if somebody smothers you or you're drowning or something, the lactic acid will build up quickly.
So
And that's bad.
fermentation is just
Yes.
the cells are getting enough oxygen, and then NADH comes and gives the pyruvates, protons, and electrons, and then that makes it lactic acid.
That's right. And the lactic acid is less toxic than pyruvate. So lactic acid can exist in your cells for an extended period of time without killing your cells.
But it doesn't have as much energy as pyruvate.
What's it like toxic?
It has energy, but pyruvate just can't sit there. So
it's less
it's
toxic
changed.
than the pyruvate.
Yes, lactic acid is less toxic than pyruvate. Let me go Steph first, and then Isaac, and then Emily.
How does that turn fruit juice into alcohol?
We're going to get to alcohol in a minute.
Now,
Isaac.
does lactate have something to do with milk, or is it just a similar name?
Lactose.
Similar name.
Like lactase.
Yeah, I don't know why that's similar. I don't know that. Yes.
Okay, I still don't understand how pyruvate turns into lactic acid.
Do you remember when we broke down the glucose and we produced pyruvate, that some electrons and protons came off and we made NADH? Well, that NADH can come back and give back electrons and protons and change the pyruvate molecules to something called lactate.
Okay.
Are the carbons
Is it when
changed?
it goes into the mitochondria then?
No, it just sits there as lactate. And it waits. Now, once enough oxygen gets into the mitochondrions, then they'll be changed back from lactic acid to pyruvate, and then the pyruvate will go into the mitochondrions.
So the what gets onto it? NADH? Or NADP? What is it?
NADH.
Is lactate still like 3 carbons? Like, does it change the carbons? Or
No, it doesn't change the number of carbons, it changes other things associated with it.
So it just adds protons and
Yes.
Is this 8.3?
This is in 8.3, yes. So we call this production of lactic acid, we call it fermentation. Fermentation is production of lactic acid, and that's what animal cells do. Animal cells do fermentation and produce lactic acid if they don't have enough oxygen. But plant cells do what's called alcoholic fermentation. Instead of producing lactic acid, the pyruvates are converted to a 2-carbon molecule called ethyl alcohol. Where not only did the NAD give back electrons and protons, but also one of the carbons comes off and floats away as CO2, carbon dioxide.
Can you do it again? It gives protons and electrons and what else?
NADH gives protons and electrons back to the structure, and carbon comes off of the pyruvate, and now we have a molecule called ethyl alcohol, also known as ethanol.
Wait, does it just bind an O2? And
No, there's already O2 in the structure. There's already oxygens connected to these. I just don't draw them in because they're it would make it too hard to understand.
Is this only when there's not enough
When they're getting some away.
Just when there's
Yes, it happens when there's not enough oxygen and the pyruvates cannot enter the mitochondrions. If there's not enough oxygen present, the pyruvates will not enter the mitochondrions, and in plant cells, the pyruvates will be converted to ethyl alcohol. The NADHs that were produced come back and give back their protons and electrons, and one of the carbons falls off and exits as carbon dioxide, and now we have a new molecule called ethyl alcohol, which is not very toxic to plant cells. So if you want to make alcohol, which you do, You simply take a plant, you put it in a big tank called a fermentation tank. Let's use barley. We take barley, we put it in the fermentation tank. We then suck all the oxygen out of the tank. And we make sure there's glucose in there, and the plant cells will absorb it. There's no oxygen present, so they'll convert the glucose into pyruvate and then into ethyl alcohol. And bam, you've got beer. Barley produces beer. You take wheat and put it in your fermentation tank, and you'll produce vodka. You take cacti and put them in your fermentation tank, and you'll produce tequila. You take corn and put it in there, you get bourbon.
Grapes.
Potatoes, you put it in there, you get vodka. Grapes, you put it in there, you get wine. Your uncle, you put him in there, he doesn't do alcoholic fermentation. He'll do lactic acid fermentation.
Why does it happen like in
He's
like
an
a
animal.
bottle of juice and everything?
Because you got it all capped up and there's no oxygen can get in there. And there's plant cells. So it could happen that way. That's not the way they normally make alcoholic beverages.
That should be a
Interesting?
lab. We should do that in a lab.
Read about that. I took a lab in college called Fermentation Lab, and we made liquor in that class. We made beer, and we drank it at the end of the semester.
Did it taste weird?
It tasted terrible. It gave you an appreciation for, real beer makers like Budweiser and such that make something that actually doesn't taste that bad. Because the ones we tasted, we could barely finish. We could barely stomach them. They were so terrible.
What kind of
4-month lab. It took us 4 months. Yeah.
what kind of thing was made to make moonshine?
Corn.
Corn.
Yeah, moonshine is a whiskey.
Why is it
Produces.
so much stronger than other alcohols?
Well, it's distilled.
Yeah, they have ways of making it stronger, and the reason why they wanted to make it so strong was to transport it in small things, so you could hide it.
Thought they made it like during the Prohibition.
Yeah, they did.
They still
I think
they
they
still
did.
make it in like college.
Yeah.
Yeah, they make it out of moons.
There was a guy at the Jaguars game with some yesterday.
You were there?
Yeah, I went to the
Is it
game.
you?
We were tailgating, and he came around and he had a bottle of moonshine. Tasted like gasoline.
So maybe it wasn't moonshine.
It might have been gas. That's what I told him. I said, "You sure this is moonshine?" And he said, "I've been drinking it all day."
Probably went blind after that.
Maybe.
What is this is kind of random.
Yes.
What is absinthe? Like how does that work?
Absinthe.
Absinthe, yeah.
Green Devil.
Oh, I don't know.
How does that work?
I don't know what that is.
You don't know what
It's got
that
some
is?
like hallucinogen in it.
Yeah.
Isn't that like illegal in
It
Europe or
makes you
something?
trip.
It's legal in America.
No, I've never done that.
I haven't either, but I'm just saying.
That happened.
Okay. Now, let's see what happens. Okay, come back to this. Let's see what happens when the pyruvate goes into the mitochondrion. And again, that will only happen if there's plenty of oxygen. But if there is plenty of oxygen, the pyruvate enters the mitochondrion. And this is 8.4. And when the pyruvate enters the mitochondrion,
Wait, the both of them?
preparatory reaction. Yeah, both of them will enter. And in reality, there's many thousands of them entering.
They're not like attached though.
Not to one another.
They're just.
They enter the mitochondrion, and what I'm going to do on this board is I'm going to make a big mitochondrion.
Wait, the
Yay.
preparatory reaction?
The preparatory reaction. Can I make a big mitochondrion? Will you all allow me to do that?
No.
I'm going to make a big mitochondrion. Here's the walls of the mitochondrion.
Some people are like,
You can't see those lines. But
And here's the membrane of the mitochondrion. Do you know what the membrane of the mitochondrion is called?
Christe.
The folds are called cristae. I remember Christy Barrow took my class. And she could remember, because it was her name. Do you see how the mitochondrion here has these folds? And actually there are two membranes. There's the inner membrane and the outer membrane. I have the inner membrane right here, and this is the outer membrane. So the folds of the inner membrane are called cristae. And there's all types of proteins scattered along the folds of the inner membrane. I ought to make this lower so we can see it a little better. Here are the proteins scattered along the inner membrane. I'm only going to put one set of proteins, but you have to imagine there's a bunch of proteins. Okay, so here comes the pyruvate. I'm just going to use one pyruvate, okay? So you don't get too freaked out. Here's one pyruvate coming into the mitochondrion. The next thing that happens is called the preparatory reaction, which is on page 142. Where's my prep reaction? Where is it? Here it is. You see page 142? The prep reaction is basically this. The pyruvate, which it shows right here, see how it's a 3-carbon molecule? C, 3 carbons. There's also some hydrogens and oxygens stuck on it, which I don't want to show you. That's okay with y'all. This 3-carbon molecule gets added to a molecule called coenzyme A. And they get added together, and one of the carbons comes off. And that carbon will exit the mitochondrion as carbon dioxide. See, this carbon actually has a couple oxygens hooked to it. And when it breaks off, now it's CO2. And that carbon dioxide floats away.
What's it called?
What happens to the that hydrogen?
I don't know. I don't know what happens to the hydrogen.
Is this just like
Let's
an
not
empty
worry about it.
space?
What's that?
It's just happening in like an empty space.
It happens in the middle of the mitochondrion. The very center of the mitochondrion is called the matrix.
I don't get why those oxygens are attached to it.
Because this molecule here that I'm showing you, pyruvate, is not just carbons. It's got oxygens and hydrogens too.
Well, if it has hydrogens, then why is it CO2?
I don't know. One of the hydrogens must fall off, and then you're just left with the oxygens.
So pretty much preparatory reaction makes carbon dioxide.
Yes. The preparatory reaction, it goes like this. Pyruvate attaches to coenzyme A, and one of the carbons falls off, and you're left with a new molecule. This molecule is called acetyl-CoA. Acetyl coenzyme A. Yes, Jonathan?
Very bad.
Oh,
That's what the new thing's
you're
called.
going to miss good stuff. I guess so.
Acetyl coenzyme A.
Acetyl-CoA.
Which is carbon dioxide?
No. This molecule is acetyl-CoA. This molecule is carbon dioxide.
Acetyl-CoA is the 2 carbons plus the coenzyme.
There you go.
Wait, I'm a little
Good.
confused. Thanks. First things over.
Ah, you've been good.
Can I ask a question?
Yes.
So, just tell me what happens once the carbon dioxide comes off.
Then you're left with acetyl-CoA and carbon dioxide.
Okay, but then when does it leave as carbon dioxide?
It just flows. This carbon dioxide can flow right through membranes, and it just diffuses out of the cell.
So the carbon that comes off is what leaves.
That's right.
Oh, okay.
And one more time, why does it come off?
Yeah, why does it
Well, what happens is we take this molecule and we break it apart. Do you remember what happens when something is broken apart?
Why is it broken apart? How?
Because the breaking apart of the molecule releases energy.
Is it unstable? Is that why it's breaking apart?
There are enzymes there breaking it apart. Good question. There are enzymes there breaking it apart. Enzymes pull this off. I'm not showing you the enzymes. Good job there, Lee. I should have explained that. But whenever a large molecule is broken apart, protons and electrons fly off, and they're picked up by NAD. So it picks up the electrons, it picks up the protons, and forms NADH. And that NADH is a high-energy molecule that will be used in just a minute in the electron transport chain.
The electrons form NADH?
The electrons attach to NAD. And so does a proton attaches to NAD, and that forms NADH.
And NADPH isn't found in this at all.
No, NADPH is in photosynthesis. NADH is in respiration.
Can NAD and FAD be used for the same things?
Yes.
Why?
No, FAD is not used here. FAD is used next in the citric acid cycle. We'll talk about that tomorrow.
What's the NADH look like?
The NADH goes to this last part, electron transport chain, which I'm going to talk to you about tomorrow. So, can you see? Look at this formula. 2 pyruvates plus 2 CoAs. See, there's actually 2 pyruvates doing this at once. And there's 2 CoAs. And so, 2 pyruvates attach to 2 CoAs and produce 2 acetyl-CoAs, that's these 2, plus 2 carbon dioxides, that's these 2, and 2 NADH are formed in the process. 2 NADs come in and pick up protons and electrons and become 2 NADHs.
Where's the citric acid cycle?
That's coming next. That also happens in the matrix.
Wait, what step is fermentation in?
Fermentation happened way out. Stepped on your model. Fermentation happened outside the mitochondrion if oxygen is not present. That's when fermentation will happen.
So it only
We're going ahead if oxygen is present, it goes into the mitochondria. So fermentation happens before this.
Mm-hmm.
So it only happens in certain situations.
That's right.
So there isn't enough oxygen.
If there isn't enough oxygen.
So the pyruvate connects to coenzyme A, and then that CO2 thing breaks off.
And then leaves carbon dioxide.
And the CO2 floats away. In our bodies, you'll breathe the CO2 out with every breath. You breathe out carbon dioxide. Do y'all remember that?
Is it strictly? Is it
20 trillion carbon dioxides are just breathed out. There's 20 trillion more. I can keep going if you need me to. What was that?
Do we only breathe out carbon dioxide?
No, you'll breathe out some water and some other things. Any types of things that don't belong in your body. Some other gases, nitrogen, some oxygen, but yeah, carbon dioxide is the main waste product that's breathed out. Do y'all understand the prep reaction?
Alright, citric acid.
Now's the citric acid cycle.
This camera's waterproof.
What?
This camera is waterproof.
Nice.
So it says.
We spare no expense at Frederica.
It's also shockproof.
Huh? Bah! No, it still works. Okay, what happens to the acetyl-CoA?
Goes to the citric acid.
Citric acid cycle, that's next. So here they're showing the mitochondrion, and they're showing you where it happens in the matrix. The matrix is the middle of the mitochondrion here. Do you remember the movie The Matrix?
Yeah.
Has nothing to do with this.
Yes.
But, you know, when they said in that movie, when they said, Neo, do you know what the matrix is? I jumped up and went, it's the middle of the mitochondrion! And I looked around and they escorted me out of the theater.
Wait, really?
No, I'm kidding.
Okay.
I was like, what is it? What's the matrix? Anyway, so the acetyl-CoA goes through what's called the citric acid cycle. And the citric acid cycle has a bunch of steps. And they show it here as this big circle thing. It's kind of like the Calvin cycle. Do you remember how the Calvin cycle had different steps?
Yeah.
You could go through and memorize all the steps, but I don't really want to waste time doing that because to me it is a waste of time. They aren't going to test you on this, and it's kind of minutia of cell dynamics, which if you're really into cells, you can learn that in college in a later course. What I'm just going to tell you is, during the citric acid cycle, these acetyl-CoA molecules are completely torn apart. During the citric acid cycle, this acetyl-CoA molecule is completely torn apart.
How was it put together then?
It came in as glucose, right?
Yeah.
So does the CO2 break when that breaks?
Came in as pyruvate.
It came into the mitochondrion as pyruvate. In the preparatory reaction, one carbon came off. In the citric acid cycle, the other carbons come off.
So it turns into glucose when the pyruvate combines with the...
No, it was glucose before it ever got into the cell.
Oh.
Plants made it. And now an animal has eaten the glucose and we got to tear it down to make energy. So we're at the very end of the breaking down of the glucose. The acetyl-CoA is completely torn apart in the citric acid cycle. And the reason why they call it citric acid cycle is this CoA will hook on to another molecule. And let's just imagine the other molecule is right here. And when the CoA hooks on to the other molecule, we form a new molecule called citric acid. And that's why they call it the citric acid cycle. But the citric acid is eventually completely broken down, and you're left with that first molecule, so another acetyl-CoA can attach and eventually be completely broken down.
Wait. When you say left with the original molecule, which original? The CoA or
this molecule? Let me remove this. What? I've lost my carbon atoms. Luckily, I have a whole thing of carbon atoms. Okay, let me go back, EKF. You have to look. The acetyl-CoAs. Whoops, I did the wrong one. The acetyl-CoAs attach to some molecule, release, and then the what's left of the carbon molecule is completely torn apart.
Yeah, and it makes.
And this thing is left there.
What is it?
And it can attach again to another acetyl-CoA, and completely it's torn apart in the citric acid cycle. So that's why it's a cycle, because this thing keeps coming back for more.
What is it though? That's citric acid.
What's the name of it? It's called oxaloacetate, if that's something you're interested in.
Okay. Well,
But you're probably not interested in it.
So we just
I'm not interested in it. All you have to say is in the citric acid cycle, the acetyl-CoA is completely torn apart.
Okay.
We'll get back to that tomorrow. So read through the citric acid cycle, and I'll start there tomorrow on 8.3. So you're to read
How do I